imaging technologies have progressed rapidly over time to keep pace with the steadily evolving fields of biological research. At present, there are techniques that let us observe biological phenomena at the nanoscale level and monitor dynamic changes in tissue networks at single-cell resolutions. However, many of these methods are complex and require massive, complicated, and expensive setups to function. To overcome the bottlenecks that exist in imaging today, the Biophotonics Research Group is spearheading innovative research at the forefront of microscopy. The group has recently conducted 4 studies aimed at improving spatial and temporal resolutions in microscopy. The first study uses a laser diode-based pulsed light sort system and time-gated fluorescence detection to develop two-photon excitation pulsed stimulated emission depletion microscopy. The researchers were successful in achieving nanometer-scale super-resolution imaging. This microscopy will facilitate a deeper resolution observation of the dynamics and mechanisms of physiological functions in living tissues and individuals at the nanoscale. In the second study, researchers used an image analysis-based super-resolution technique, SURF, in combination with two-photon microscopy for deep imaging. The technique, called TP-SURF, led to the successful high-resolution imaging of neuronal dendritic spines and other minute structures in fixed mouse brain slices and also in the in vivo mouse brain. This method can be easily applied to existing two-photon microscopes and is expected to maximize the range of applications for super-resolution imaging. In the third study, the researchers developed an easy-to-use light needle creating device for imaging thick tissue specimens. This system demonstrated its usability in visualizing the 3D structure of transparent YFP-expressing brain slices. The team could also detect calcium activities with a high temporal resolution in mouse cortical slices. In the fourth study, researchers developed a novel fluoropolymer nanosheet called polyethylene oxide-coated Cytop (PEO Cytop) nanosheets. The researchers performed in vivo deep imaging on mouse brains at high resolution and used the PEO Cytop nanosheets as the sealing material. The findings showed the potential of this method for investigating neural functions involving interactions between multiple brain regions. The highly advanced microscopy techniques developed by these researchers will help better understand brain function and improve diagnosis and treatment of mental disorders.